Okay, well, it says we're live again. Oh, I'm getting red again.
Yes, the
Okay.
internet is red again.
Yep,
Yes.
as soon as we went live, it went red. Okay. Whoop, and it's fixing itself. All right. Okay, well, hello, everyone. We're back. Probably, maybe. We are going to see how this works, if this will work. Because my internet died on me, as it is wont to do sometimes, probably creationists in the wires. And so we'll just pick up where we left off until such time as it drops again. So, uh, welcome, Dr. Buzgo. Welcome back. How you doing?
I'm doing fine and trying to remember where we were. So
We were talking about archaeans and
we made a
endosymbiosis.
huge jump in the origin of life. Made a huge leap from chemical evolution. Now we're already middle of Archeans, discussing about the two domains of prokaryote life. So you can say the two principles of life. The Archeans was bacteria. Yes.
Right, which is kind of funny because the you know the origin of eukaryotes is about halfway through the entire history of life itself. From about 4 billion years ago to 2 billion years ago, and then you have the other 2 billion years to today. So that's just about right the middle. So we kind of jumped, which because
Yes.
we don't think about it in terms of scale, but half of the history of life was no more complex than bacteria. So
Exactly.
yeah, it's crazy.
So eukaryotes, organisms with cells with nuclei and complex organelles inside, they exist over half the time. And half of the other time, we didn't even sit around. It gets even better when we think about oxygen atmosphere, which is only about a third, less of the time that we really have solid oxygen in the atmosphere. And then, of course, we move all the way to where we have life forms that we can see macroscopically that make some impression, literally. So the Cambrian, Ediacaran times, we talk about something, the Cambrian is a 500, and what is it, 510 or 570 million years
For
ago.
the Cambrian the start of the Cambrian was 540 million years ago. Yeah.
Yeah, so 540 years ago, so the explosion just happening basically recently. So anything we know about this planet happened relatively recently, is the exception of a very drab, deserted planet, which has nothing more exciting than bacteria. If you're an alien and you land at any random time on Earth between its existence and disappearance, half of or today, then 50% chance you meet only bacteria, and then more than that, like 75% chance you meet only single-celled organisms that don't talk about existentialism with you. And so then you just leave again and look for a planet that's more exciting. And I think if we look at life in outer space and go out and try to find life in outer space, we may find life in outer space. We're going to be terribly disappointed because they don't have flirtatious relationships with us like in all these sci-fi movies we watch. They're just sitting there metabolizing quietly and doing nothing else. That's going to be quite disappointing. Yeah. So we are now in our kids. We talked about how eukaryotes came to life. Let's say this is a different story, but the big thing about eukaryotes that I find is actually the formation of the entire chromosome container, the nuclear membrane, and then the spindle apparatus and microtubule system. While the invasion of the mitochondria, although quite important and very often praised as a great invention, might have been a secondary impact or something that nice to have, but it's not necessarily making the eukaryote, although all of them have mitochondria, but it's not necessarily what makes you what you are in that sense. There's a lot of other things in it which sometimes slip out of textbooks. People don't think about that. So, but
Right.
also,
Yeah.
how did that happen? How did these eukaryotes actually come to place?
Do you think that I know Nick Lane posits the hypothesis, I'm sure he's not the only one, that the nuclear envelope was a response to the mitochondria because it's giving off things uh, like things it uses for PCD now. Uh, like I can't remember any of the names of the enzymes, but it's giving off different enzymes which would cause, cell death. Uh, and having an envelope between the the Archaeans' genetic material and the stuff given off by the mitochondria might have uh, been an adaptive advantage. What do you think about that?
That's a speculation. The mitochondria well, I mean, no, the mitochondria form free radicals which can damage the DNA, but there's
Right.
other reasons to form a nuclear envelope. One of the key reasons is that a lot of enzymes, also those that manage part of the DNA, are membrane stuck. They're bound to membranes. And as your cell increases in size, you get a conflict between placing your DNA, your chromosomes, near a membrane, which would be cell membrane in the periphery, and having your DNA available for transcription without cell skeleton, cytoskeleton, then move those transcription molecules around easily. So the best place for DNA would be in the middle of the cell, but for processing it, you need to have it temporarily at the membrane. So one solution to keep your DNA in the middle of the cell and yet have it close to a membrane that can handle it is to pouch your membrane in from the cell membrane and form basically a access pouch, or like an access road from a ring road to a center, to access this and have this DNA then bound to a membrane while sitting in the middle. And that is exactly what actually the nuclear membrane would be, including the endoplasmatic reticulum. That explains two things. Number one, why is the nuclear membrane a double membrane? It's a basically double bubble around you. And why is the endoplasmatic reticulum directly connected to it? And that makes a lot of sense that these membrane tunnels that form from outside, of course, they're hollow inside their tunnels, and they explain what the endoplasmatic reticulum is, and they also explain to you why this cell membrane is a double pouch, is a double membrane pouch. And so yes, the mitochondria may or may not have played a role in it, but there certainly are other reasons why you would form a nuclear membrane around a more and more complex chromosome. Keep also in mind another observation. A lot of eukaryotes, they do not dissolve the nuclear membrane when they do the M cycle, the M phase. So when they pull chromosomes apart for cell division, all that stuff, so fungi, diatom algae, brown algae, dinoflagellates, a bunch of other organisms, they don't rip their nuclear membrane apart in the prophase.
Right.
And that makes a lot of sense. For example, if you're a fungus and you have a lot of nuclei in one cell, you would dissolve the cell nucleus, then you get a total salad of chromosomes in your cell, and then you can't do the spindle apparatus will not pull your chromosomes nicely sorted into one side and the other side. It would create a complete mess.
It'd just be spaghetti. Yeah.
Yeah. I mean, this would be a total salad. And so you want to keep your chromosomes while you pull the chromatids apart inside of a container so you don't mess them up, you don't jumble them. So membrane, a nuclear membrane can have a lot of reasons other than as a response to mitochondria. I don't say it's not, it's just that's not the only explanation. Sure. And as I say, it's rather speculation. You can speculate things when the day is long. So
yeah, we just can't, we can't take our nice handy dandy electron microscope back there and take a bit of seawater and just look through it, unfortunately.
No. And the other thing we need to keep in mind that these organisms are possibly quite diverse.
Sure. Yeah.
Look at evolution, we see that a lot of organisms are quite diverse, and then most of them died out and one survived. Mm-hmm. Think about the different fish that existed and how few of them actually survived. How many of those stegocephalian amphibia existed when the amphibia came out of water, became tetrapods, and all of them have died out. How many different bird-like dinosaurs exist? Several of them being able to fly and have nothing to do with birds directly. Like things like Macroraptor was not a bird, but it could fly with feathers. So there was a lot of this diversity forming, and then few survive. And so if you would go back with a time machine and look at the cells, you might have maybe a problem to find actually which one is the ancestor of the eukaryotes and which one is just a side branch doing something very similar.
Yeah, and
that's.
Yeah, I've actually been reading. I'm almost done with Wonderful Life by Stephen Jay Gould, and he kind of talks about how the history of life is what he calls bottom heavy. You have this large degree of experimentation and then restriction, and then you go from there.
Mm-hmm. Right.
And so,
while we can see this in bones, when we have bone fossils, or also plants. Some of the plants do also crazy stuff. And you can see this in the fossils. We don't have the fossils of the prokaryote single cells, so we are left a little bit in the dark about this. But if you take a time machine, go back, you might actually see exactly this Goldingian bottom heaviness of evolution there too. So I think at this time we just have to leave it at this until we see more evidence, and we can speculate all over what we want to do. For me, some of the key parts for knowing more is, as I said, spindle apparatus, or anything that points towards a sophisticated cell, a cytoskeleton, or something that goes in the direction of open chromosomes with telomeres and the centromeres in the middle. So anything like this is interesting, and it's absolutely possible that someday we find some kind of archaeans that have one of those features, at least in the pre-stage.
It's funny you mention that, because there is a genus of archaean with cell compartmentalization, and there is a clade of bacteria with cell compartmentalization. The archaean.
But
go ahead.
Compartmentalization is very convergent. Yes.
Yes. Yeah. Absolutely.
Yeah. I mean, the crystine in mitochondria and the thylakoids in cyanobacteria and chloroplasts are not the same.
Right.
So and then think about that the rhodobacter, which are a group of photosynthetic alpha proteobacteria, have also compartmentalization, very similar to what you see in cyanobacteria. Again, the compartmentalization of a cell itself is not. I'm pointing directly to things like microtubuli.
Right, well.
And actin filaments. That's where it gets fun. Or chromosomes that are not ring-shaped, but steady open, and having a distinct grouping of centromeres and telomeres. Anything in that direction, that's getting cool. And if you find some archaeans that have something like this,
I wouldn't be surprised, yeah.
That would be interesting. Compartmentalization of the cell can happen. That has happened several times convergently. We've seen this, so that doesn't wake me up on a Sunday morning.
Oh, I mean, I think it's kind of interesting. I think it's the other kind of really interesting thing about unicellular organisms is looking at, say, cyanobacteria. You look at things like the aconites and the heterocysts of guys like Nostoc, where it's almost this kind of cell differentiation within these filaments, which seems kind of reminiscent of...
We're still talking about the beginning of life, right? We're not going to multicellular organisms in the same presentation.
I was just thinking...
way far ahead. Way out of your video topic right now.
Well, yes, at the moment. I was just making a point about how that interest... I find that very interesting, because you're saying it's...
Oh yes. You have several bacteria flirt with the idea of multicellularity, and cyanobacteria are particularly complex bacteria. They are not simpletons. So yeah, they have aconites and they have heterocysts, and then you have spore formation in several bacteria. You have work compartmentalization in some bacteria. It takes enough genes to make that complex operation, but apparently cyanobacteria have quite a powerful, very diversified genome. But that's a completely different story then. We're as we say, we talk about the beginning of life, and we can maybe enter the formation of first prokaryotes as the end point of the beginning of life. But if we go beyond that and then discuss the evolution of cyanobacteria, there it gets really complex, and that is a much more sophisticated, and then we talk again 2 billion years into life already, or at least 1 billion years into life. So 3.5 billion years ago we had the first cyanobacteria that looked like oscillatorians. So we're way ahead. So you definitely did not go to the beginning of life and end up with cyanobacteria next morning.
Right.
They are way ahead of that. They're way more complicated. One of the topics that we could ask us is, which are the most simpletons among the prokaryotes? So which are the most simple, most primitive archaeans, which are the most primitive bacteria. And I think that is a more interesting question in that sense.
The off the top of my head, I'm not too in depth on bacteria systematics, but from what I understand, there's a dichotomy between the hydrobacteria and the pterobacteria, but of which in those groups is closest to the base. I'm not sure exactly what the consensus is, if there is a consensus on it.
Yeah. It's a bit hard because there's a lot of horizontal gene transfer, but the bacteria phylogeny, as I have it in my head, one of the first dichotomies is between gram-positives and gram-negatives, between the Firmicutes, Dendricutes line, and then the entire rest of bacteria.
Yeah, everybody else.
Yeah, then everybody else. And so that is already a big point, because all gram-negatives have a double membrane, originally at least.
Mm-hmm.
And the gram-positives, the Firmicutes, don't. So which one was first, double membrane and single membrane? And you have a double membrane. How did that come to existence? What is the outer membrane versus the inner membrane? Which one is the original one?
Right.
With the secondary one. A very heretic proposal here, which I have no evidence at all, just heretic. Who tells me that not the bacteria themselves are a endosymbiont situation, where the inner membrane of the gram-negatives is actually a secondary invader into an earlier existing outer membrane forming prokaryote.
Like a diatom on top of an algae sort of situation, right?
Or
got-
a mitochondrium inside of an archaean.
Right, yeah, you've got like endosymbiosis inside of endosymbiosis inside of endosymbiosis.
So imagine yourself if one day we discover that the outer membrane is an archaean membrane and the inner membrane is a bacterial membrane. And what would that tell you about the origin of life or the origin of prokaryotes?
Everything's just an archaean.
And everything is just an archaean, and the archaeans are basically having then, or the archaeans, they kind of engulfed bacteria again. And so in that part is we don't know where the double membrane comes from. How did that evolve? And as long as we're tapping in the dark like this, we don't even know whether they are the gram-positives or the gram-negatives represent the ancestral character state of the bacteria clade, of the bacteria domain. All is just speculation.
Right.
So I just would like to see more evidence before I go in and speculate whether
but where's the fun in that, right? Well, since we I don't want to be here I don't know how long you want to do this, but we're not to the hour mark yet. We're about 45 minutes or so is how long we've been doing it. But let's see. Let's say we cross the eukaryote barrier now. So we've got an archaean who takes in. So we're about 2 billion years ago, and that about how long after the endosymbiotic event do you think it was before the three major modern clades separated?
3 domains.
Yeah.
Or the 3 major clades of
eukaryotes.
But are there 3 major clades or only 2?
Well, if Biconta and Excavata are actually diaphoretics or whatever, then it would just be 2, I guess.
Yes.
Do you have an opinion on that?
Yeah, I mean, Excavata have 2 membranes, 2 flagella.
Right.
Just that one behaves different than the other one. So in Euglena, one is very short, one is very long. In Trypanosoma, one forms like a dorsal fin, and it's inside of a longer fused membrane rim inside of the cell, and forms like a dorsal fin, while the other one is free flagelling. More or less, it looks more like a tapering tail. And I don't even know if they're interchangeable, if they can flip them. I don't even know that. But Giardia has clearly 2 flagella. Giardia is the digestive issue. It gives you 2 weeks of normality, 1 week of constipation, 1 week of diarrhea. So a nice clockwork of 4 weeks. And that's a reason to wash our hands after the toilet, because that's where God travels, okay? Think about that. And so Giardia has clearly 2 flagella. And I'm not an excavata expert at all. I just know those from the textbooks. And if you dig deeper, I don't know if any excavatant has really only 1 flagellum. And even if a flagellum lost or reduced is easier than to build one, you easily lose a flagellum. I mean, some organisms have no flagella at all, lost all of them, like red algae, for example. So I think the excavata, just from the cell shape, they pattern very nicely into the Biconta group, and then there's only 2 groups of life. Now, which one is earlier, I don't know, but I could always imagine that the Biconta were earlier and the Uniconta split off from them by losing a flagella. Mm-hmm. That's just a parsimonious approach of speculation here. Now, I don't have the most recent DNA analysis. All DNA analysis I've seen, so far, they had, with support, those that have supported branches have a bifurcation in Uniconta and Biconta, and the excavata having a poor support.
Mm-hmm. Yeah, it's
being polyphyletic with the Biconta, being polyphyletic at the base. So that's where you talk about the clades, but we don't even know it's only 2. It's only 2 clades, and so we actually don't live for 3 clades.
Right. Yeah, that's part. It seems like every possible topology has been given for excavata, whether they're they're monophyletic and they're at the base, or they're paraphyletic or polyphyletic, or they're next to Biconta. It seems like every single possible one has been given at some point, because they just kind of keep floating around.
But I think this is one of the problems when you rely too much on DNA analysis only, and the DNA phylogeny. You have 2 problems pop up. One is very long branches, very far isolated groups that are separated, pattern into 1 common branch, long branch attraction. And the other problem that you have is that with the case of excavata, you have a lot of horizontal gene transfer going on, and sometimes messy sexual reproduction cycles. A lot of the euglenophytes are not having any sexual reproduction known, or uncertain sexual reproduction. So parasexual. And so we don't quite know what they're doing sexually and how gene recombination happens over a long time. Now, if these guys have been around for a long time and have had some endosymbiosis going on with secondary inheritance of chloroplasts, it can give you a hint on how reticulated evolution can be. So maybe DNA will not tell us. Maybe we will have to find some other data at some time, more in the functional side, more in the ribosome side, or something like this. So ribosome function beyond the sequence, it can give us some extra hints, or membrane compounds, membrane pattern that we have not yet identified as being diagnostic. Maybe there is more information in that, but at this time, we're just stuck with what the DNA analysis tells us, and apparently it's inconclusive at this time.
Right. Regarding the excavata, one of the most interesting stories that you ever told us was regarding this group and how phylogenetics actually saved somebody's life, because there's
yes,
one of the common things I see, and it's not just from creationists, it's from kind of common people. It's like, well, is evolution? Can you use it practically? Is it just data gathering, or is there a use for it? And
is it just art for art, or is there an application for it?
Right, and the example, which, if you want to tell, I think is a very good example of that.
Yes. Again, what was her name again?
Kelly Hardig.
Kelly Hardig. Yeah, she was a girl who contracted what's known as brain-eating amoeba in the public. It's a little excavate, I think a kinetoplastid flagellate that lives in warm water, and they're just flagellating around eating garbage. And it can happen that people that who get that little critter up their nose from swimming, diving, jumping, splashing water around. And this critter attaches itself to the odor nerve cells where we smell with and slip into those nerves and thereby dodge the immune system, then creep inside of those nerve cells right from the nose into the brain. Now, people who have looked at human skulls, they see that actually between the roof of the nasal space and the brain cavity, there is a little thin bone with little holes in it, like a coffee sieve. And these holes are actually where the nerve cells go through that connect the brain, the bulbus olfactorius, directly with olfactory nerves in the nose. It's a very ancient nerve system. And it's a nerve system where the brain nerves go directly to the outside into our nose. And then these flagellates go into the brain and start basically there to kill the cells and to eat them. That liquidizes the brain and from the front. And people who contract this disease, they first feel bit nausea, maybe visual problems, maybe not. It feels a little bit like an oncoming flu or headache. And before you know it, it's too late, the person is in coma, and in that case, it's basically 100% death rate. So who gets it dies. No rescue, you're lost. And so in this situation, Hardik, she was the parents aware of it. She that in Arkansas, a little swim corner on the river where lots of people have been swimming for years. Nobody got that there. And she got that and contracted. So the parents were very alert, reacted very quickly, took her to the hospital. And then the hospital realized very quickly what this was. And the first thing they did, they put her into artificial coma and cooled her down. They basically cooled the body temperature down, which is known to delay brain damage. It's also used in operations when people go into brain surgery. They cool you down so the brain damage is less in this procedure. They bought themselves a bit of time. By doing this, they slowed down the decay of the brain, and they bought themselves some time and could, in that time, import a new drug, a new medicament from out of state. It was developed at a university not to treat that particular flagellate, that Naegleria fowleri that we talk about here, which is the cause of the primary amoeboid meningitis that we talk about, but it was developed to treat a completely different disease, Chagas, which is related to sleep disease. Chagas is basically the American version of sleep disease.
Right. Poor Darwin.
And,
I said, poor Darwin. Yeah.
Yeah. And so the reason they thought that this medication could help is because Chagas is also a kinetoplastid. It's also an excavata member, just like Naegleria fowleri. And he did. And so Hardik is alive. She had to go to rehab. She had some brain damage from this, of course, but she's young. The brain could remap, and she has gone into rehab. And from what I know, she has recovered pretty far. I mean, she gives interviews, she talks about her experience, and it saved her life. And she was first one that they tried the same medication in other cases, in some cases with some success, in many cases still not. But that idea that one medication helps to another organism that's closely related, that makes a lot of sense. It also operates in a much easier way. For example, mites. Those little spiders that can live in your mattress. Those spiders, they live in your skin, give you an itchy skin disease. Other of these mites live in honeybees and kill our honeybees. The big colony collapse is related to a mite called Varia destructor, or Jacobsoni is mostly the destructor species. And the same poison that works on a pet mattress, mites on your skin, can also work at mites on honeybees. And of course, now you can ask yourself, why would it do that? Well, they're all mites. It makes sense, right? But why are these mites all mites? What combines them? The thing is that they not only look alike, they also have their internal body structure, their entire chemistry alike, and that's why these medications work.
Right.
That is, I think, the key part why phylogeny helps us to understand it. Now, we talk about mites, we understand why this works. But if you talk about single-celled organisms or bacteria, it gets much more complicated, and there the phylogeny can really be saving life. The same also with viruses. Think about coronavirus. Next relative coronavirus that scared us was SARS.
Mm-hmm.
Both coronaviruses, and they both behave similar and might both respond to the same drugs as medication. Viruses, the same medication helps against herpes, shingles, may also work against chickenpox and other herpesviridae. So phylogenies work a big deal. Also then with cancer cells. Cancer cells mutate. If you take chemotherapy, by default, you kill 99.9% of the cells. All cells that are susceptible to your chemotherapy will die. But there's 1 or 2 cells that are not susceptible, they will not die, and they will then cause the relapse after you've gone into remission for a while. So if you can trace down that the second cancer, the relapse of the cancer, so to speak, is actually the descendant of the first cancer, you can save yourself some chemo because you know that's exactly what they survived. You don't have to apply that chemotherapy anymore because that's what they survived. If it's, however, a different cancer that has not been created by survivors of the first cancer attack, then the same chemotherapy might actually be useful again. And for people who have taken chemotherapy, it's stressful, it has a lot of impact, and you don't want to go through chemotherapies that don't help. So also think it all day. Time matters. You don't want to try chemotherapies that don't work on you and lose time on it. So you want to be more efficient. So phylogeny actually does save lives. Oh yes, and it is very useful. Yes.
Yep. So it's right. It's not just art for the sake of art. It does have actual practical application.
And it's not just kind of running after some fancy-looking orchids. It is saving lives directly.
Yeah. Exactly. Take that, creationist.
It's not just some orchid species discussion. No, it saves lives. Yeah.
Moving on down because I do want to finish on plants because that is your.
No. Now we are beginning of. Remember, beginning of life was the agenda topic we're still discussing.
Well, that was the first topic. We talked about origin of life. We kind of went through procreation.
I thought you wanted to talk through all the topics. Okay.
Well, yeah, I wanted to go through. I wanted to kind of hit
all of them.
Well, at least some of them.
Like, that's good. We're done with the plants. Okay, we're okay. We know you talked about phylogeny. Now we talked about the value of phylogeny and Harding. Yeah. So now we check that off. Okay. Now we move on to the next level.
Yeah.
What's on the? I don't have it on my computer. I didn't print it out. Help me.
Oh, you're fine. Yeah, I've got it here on my screen. So I do want to end on plants. So it's going to go down the Uniconta. So as we are uniconts, because one flagella unicont, and so it's kind of interesting that fungi are in our group. They're not in the plant group, and so are amoebas.
Take that, vegans. They're animals. Fungi are close to animals. Think about that if you eat garden mushrooms. You're not breaking your rules. Yeah. Careful with that.
Yeah. It's very interesting, and they keep discovering more and more little groups of protists kind of along the line between amoeba and fungi, and between fungi and animals, and we keep kind of expanding the repertoire of genes that were thought to be animal specific, but it turns out they actually go back hundreds of millions of years before animals. Things like
Yes. Well, that's one of the reasons why yeast was used for understanding the cell cycle. Much of what we know about cell cycles, the entire M phase, the entire what regulates the beginning of chromosome replication, and then the reorganization of chromosomes as they get pulled apart in the M phase, we know that from yeast. And the two major yeasts that were used for that is the Schistosaccharomyces, the fission yeasts. The favorite one is Schistosaccharomyces pombe, because in Swahili, pombe means drunk, beer. And it's the yeast that was used to make African corn beer, or in the old time millet beer. And I had the privilege to try that.
Is that the one you left your colleagues with?
Yes, yes. I'm not going to name her name because you might actually be watching this. So I can give you a flavor. So we were in Cameroon and they prepared that beer, and it was not the light beer. You don't imagine your IPA. It was more the thickness of a porridge, thick, yogurty, and the flavor was a mix between yogurt and baby vomit.
Oh man!
It is nutritious, rich in nutrients, I guess, and quickly saturating adjusted to the flavor. And yes, my colleague, she took an entire bottle out of it. He was all happy and excited and had to take an entire bottle. I saw in the somewhat puzzled view of the lady who sells that beer that people locally don't do that. But what the heck? She sold her a bottle full of that stuff, and as we arrived at the headquarters, she stored that on a warm shelf, and the bottle exploded. I had seen that forthcoming, and I started to wrap up my sleeping bag and move my backpack out of that room. Wait until the big pop went off and people had loud, frustrated voices, and I knew now the bottle has popped and spread all the content around the room. Yes, and the thing is, of course, then the other one is baker's yeast that was used extensively. It's an extreme workhorse for cell biology. It's used for so many applications. And it has been used intensively to study cell cycles, also because baker's yeast, all yeasts actually, ironically, most yeasts that I know about, ironically, don't do what fungi do. Most fungi have a cell cycle that's called zygotic. That means they have almost no diploid phase. Their diploid phase is only the zygote. And as soon as this zygote reaches a certain point, it will do meiosis and break into a haploid stage again. And yeasts are among those few fungi that broke free of that contraptment of a zygotic meiosis type, and they can do sporic meiosis type. That means they can divide while being diploid. They find a way out of that. That's important to understand animal cells. We animals have a diploid life cycle. You need to see what enables a cell to suppress meiosis when you diploid. That's actually what happens. If you're normal eukaryote, you're a diploid zygote, the next division you're going to do is going to be meiosis.
So do you think that's a matter of heterochrony? Or the expression of that?
Yeah, the expression might be applying. I mean, heterochrony is rather a phenotypical description. What happens is a change of time. What it is enabling the chromosomes, when they're condensed, not to pair up. So it must cover the chromatin of chromosomes, or it's my speculation, it might cover the chromatin of chromosomes in such a form that they cannot form that homologous pairing according to the chromatin line that they do in a normal meiosis during the synapses of the prophase. And that requires a mechanism to prevent that pairing from happening. So the chromosomes float then a single shaped little spaghettis around, and you can attach your microtube both sides to the centromere and pull the chromatins, the chromatids apart, the two halves of the chromosome, so to speak, two sausages. And that happens naturally when you're haploid, because if you're haploid, each chromosome is alone and there's no pairing. But when you're diploid, this pairing naturally happens. You need to suppress that. So you can call this heterochrony if you want. For me, it is more a DNA management issue that you need to develop something that stops that pairing and that allows each chromosome to get two microtubally attaching to the centromere. It might need more steps than just covering absolutely don't pair. It might take more steps than that. It might not be just one single feature. It might be a multi-factor thing that you need to evolve to be able to suppress meiosis while diploid. But the yeast cells have done that. Yeast can do that, and so they serve as a model organism how that happened then at the transition into animals, which differ from their next relatives also by being diploid. While they're most, at least those relatives that we know, sexual cycle of, I think it's one or two species of monoflagellates that we know.
Yeah, it's not very many.
They are not gametic like animals.
Right.
Sporic, I don't know what the other one is, but definitely one is sporic. It seems that animals are primarily simply a form of gametic monoflagellates. And that seems to be here also the key innovation. And to understand animal cells or human cells, you need to understand yeast.
Yeah, and it's all.
We got fungi.
Yeah, yeah, using fungi and unicellular protists to understand the animals, because it's all a lot of the parts are the same. A lot of the genetic systems are the same, that kind of stuff, and so that's why we can even do these kinds of comparisons, because they all share a common ancestor who had these systems.
And of course, there's my problem when I teach biology and I open a textbook, and the textbook talks about kingdoms and has a kingdom of Protista. And these protists are totally paraphyletic. And there's no such thing as a kingdom of protists.
Right.
All kingdoms or supergroups, how we call them today, start as protists and then branch out, including some multicellular organisms. So the ancestors of animals were protists. The ancestors of were protists. So that word protist is actually a bad word. It's simply the base group of eukaryotes where each of the different lineages came from, and everybody started as a single-celled, every line started as a single-celled organism. And the single-celled ancestors of animals, the monoflagellates, are surprisingly primitive as compared to other protists. Right. So the common ancestor for fungi, the chytrids, this is the stem group where fungi came from, they have some things in common with the monoflagellates, like one single flagellum that's on the backside of the swimming direction. So you swim a little bit like a tadpole and not with something that has a front propeller. And that single rear flagellum is what unites fungi and animals of flagellates as Mitochondria, while the other unicorns, the amoebozoans and slime molds, they have a different pattern. They form a second group to them. So yes, there's tons of things to be discovered. And most of these are little boring flagellates nobody studies. We don't get any funding to study them because what is it good for?
Yeah,
there's always the question about this. Why is it good for? Do you want some like one is the, I think the cryptomycetes or something like this. This is one of those side branches that was discovered in the scum of a school pond by EPCR, so environmental PCR. We basically took a spoon of pool scum and threw it in the PCR machine and found some sequences that were not supposed to be there, that didn't fit for anything, and looked at them in fluorescent marking, and discovered some unknown flagellates having those fluorescent signals on them. And then you go, WTF, what's that flagellate? And so then they discovered a complete new branch between animals and fungi, and I think they're called cryptomycetes, actually, hidden fungi.
I'm familiar with, you say fungi branch off, then you got the ichthyosporians or mesomycetozoans, whatever they call them, and then philisterians, and they made another group not too long ago.
They have changed the name. They may even have changed the name. Whenever I go to Wikipedia, to change the name again.
Oh yeah, because there are like 10 different names for each of these clades, because the clades have been around for 50 years, and so every time they redo it, they got to change. And the
phytocode crazies, they go and give every node in the phylogram a new name. Yes, you can do that. If you say that every taxon needs to be monophyletic, they'd go around saying every monophyletic group is a taxon.
Right.
So you have a total inflation of taxonames which nobody can understand. Every node you give a new name, and whoever writes in Wikipedia defines his own name and messes everything up. Good luck with that. But yeah, there's a lot of groups between the fungi and the animals that keep being discovered gradually. But
Splitters on steroids.
Hmm?
The splitters on steroids.
Splitters on steroids, yeah. And I think the quinoflagellates, they are a very interesting group because they are basically like what sponge cells are, and sponges are animals. So in animal sponges at the base, and quinoflagellates, they don't really have a whole lot of difference. They don't really differ from sponges a lot. So yeah, the original animals in quinoflagellates, that is a quite interesting thing because it's a research field that you can delimit. Different what we do in the interview, where we go through the entire 5 billion years of evolution in 5 minutes.
Well, people have
you can.
Sorry.
It's a contained number of questions that you can ask and test and hypothesis and do experiments on without losing yourself in the entire life biosphere that you want to look at. You want to have questions you can delimit so you can answer them actually and dissolve the bigger questions you have in that context. And I think quinoflagellates are very, very interesting topic, and I hope that more people do research on quinoflagellates. It's really a very interesting topic, and we need to know need more about it, particularly also because sponges have been among the oldest fossils.
Mm-hmm.
If you think about the Ediacaran time, that's the time before the Cambrian explosion. We do have some fossils. Many of them are footprints of animals we don't know how they looked like. Somebody tip-tapped over the prehistoric ocean ground and let tiny little pencil footprints in the mud, and that petrified, and we say, oh, look, somebody walked across it, but I have no clue who that was.
Right.
And these have nice feet. They may have had some bristles or stalks, kind of like, as you would say, earthworms on stilts or something like that. So we don't know what they look like. And then we have some funny imprints like Dixonia. There's just this blob jellyfish that left some imprints. And yeah, we can speculate what that was. But sponges, they have tiny skeletal needles in their body build that hold them together. And these skeletal needles are diagnostic to which group of sponges you belong to. And they leave fossils as well, and quite a lot of them. So although we don't know what exact shape the sponge had, we can see that group of sponge did exist already at that time, before the Cambrian explosion. And so sponges are very old animals with an old fossil record, and therefore being basically just a sophisticated form of quinoflagellate, which is quinoflagellate on steroids. That is a very helpful bridge to tap into how old this evolved. How old are animals actually?
Yeah, definitely. I think the estimates for the last universal common ancestor of animals is about 800 million years ago.
Mm-hmm.
And then, yeah, so it's like 300 million years passed between the origin of animals and the Cambrian explosion,
Yeah, the first fossils we have, we can pin down, yeah. And with fungi, they were a bit more lucky. They found some pre-Cambrian fungi recently, and I haven't looked into that recently. But those fungi, they already have filaments. So that takes then the stem group, like chytrids, even further back. So that's always interesting when you find fossils of sophisticated organisms at a time before your expectation.
Yeah.
That puts in the beginning much further back. I bring always the example, imagine you're now looking for Nephritidis grave. Nephritidis is supposed to be in a grave chamber behind Tutankhamun, and they're looking at this. So imagine they find Nephritidis and they ask themselves about the origin of telephony. And they think about Alpha Edison with this funny spindle telephone where you spin around on a little crank and you talk into a funny tube. And they think that was the first telephone that was somewhere at 1900. And there she is, Nephritidis, with her Egyptian iconized cell phone, a BlackBerry, with little Egyptian icons on it, and some kind of copper display like the Antikythera display on it and think, wait a minute, we need to rewrite the story of our telephone invention. It was not Edison. The Egyptians already had blackberries. So in that sense, then we would have to kind of sit back and scratch our head quite a bit. And that happens in evolution sometimes. Did you find a sophisticated organism way back before you thought that's where they originated? You know, that was not the first one, yeah.
Yeah, I think I think all three. I remember reading one paper that said all three major groups of fungi, the Ascomycota, Basidiomycota, and Glomeromycota, I believe had all split by like 600 million years ago, if I remember correctly. So yeah, way back. So still Precambrian.
And of course, a person like me asks, what did this Basidium and Ascomycetes live on? Because today, most Ascomycetes live actually on land. And there's a strong correlation between Dicaria fungi like Ascomycetes, Basidiomycetes, and Glomeromycetes and plants. So if you're a Glomeromycete, you hang around in the Edicarian time, what do you live on?
Right.
Well, say maybe first land plants were around already.
Yeah, possible, I suppose. I think the earliest date to 470 million years ago, which is like a Ordovician.
Yeah, the Ordovician is the track of the oldest moss and hepatophyte, like embryophytes, yes. That we have them, but their spores,
right.
We don't know to whom they belong, older. The actual plant fossils come from the Ordovician time. We have, that's the spores that look like plant spores. They're older. We just don't know to whom they belong. And as you can imagine, mosses particularly have extremely small spores.
Mm-hmm.
So in a little hair moss capsule can have over 1 million of spores. You can imagine how small they are. So if you find fossil spores, there's not a lot they can tell you. They can say, well, I am possibly a land plant spore, maybe. But I don't tell you what they're coming from, what that land plant looks like. So we're not quite sure what happened there. So again, spores don't tell you the story. You need to have the plant. And if we have fungi that old, so sophisticated.
Well, it does seem that fungi can form relationships with cyanobacteria, things like geosiphon, which is so maybe they were having that sort of relationship pre-plant.
Lichens, yeah. Lichens have evolved several times independently, so it might absolutely be that we have some Edicarian lichen-like organisms, the fungi associated with some kind of green algae. Green algae did exist in that time.
Right.
Absolutely. Yes. Can speculate about this, yes. Again, fossil. I can speculate all day long.
Well, we're dealing with lots of soft, probably fairly small organisms and, not hugely conducive to fossilization, sadly. But yeah, we got to get our time machine and go back to the Precambrian seafloor, see what's going on.
I mean, the thing is, it's a long time. It's a difference if you find a fossil from 60 million years ago or 600 million years ago.
Mm.
I mean, 60 million years ago is only 10%.
Right.
And the fossil has survived since. If you find a T-Rex bone, we're all excited. Well, that T-Rex bone survived for 60 million years through erosion and all that stuff. Now make that 10 times longer with erosion for 10 times as long. Things that fossilized there, they might have been around at the Cambrian explosion and be already as old as our T-Rex bone. They might have been already 60 million year old fossils at the Cambrian explosion.
Mm-hmm.
Now, do that 10 more times or 9 more times, and then you reach the humans. The probability of those fossils to survive, and even if they fossilize, is very low. I think that's one of the key problems as well. Extremely old fossils are very, very hard to survive. And that is not hard. Not just that they're soft. Actually, soft lichens are quite solid. They're quite hard.
Mm.
Pretty long-living remains. So some of the lichen I find, these spore carps of the fungi, this apothecia, they're pretty hard. And I'm very sure we find fossils of those at the time of the dinosaurs, but that is nothing compared to the time of the Precambrian time. So that's our problem. Again, the time scale.
Right.
That's
Yeah.
something which kills a lot of fossils, takes them out from existence. That erosion and weather and all that stuff, and crushing rocks in the metamorphic formation that takes the fossils out. If you think, if you get yourself some marble for your countertop in the kitchen, you buy the nice one that has this little black marbling in it. That's organic material. These were plant leaves at some time, or even animals. It got smeared off in the metamorphization of that. But those fossils are not be recognized anymore.
No.
We'll never know.
But it looks nice, and that's what matters.
Yes, but this might have been the first flowering plant. This might have been the fossil of the first flowering plant that we so desperately look for.
Right.
And now it's smeared up.
Well,
Since 10 million years, it's only a black smear in a limestone rock, and it's no longer being recognized. That's what happened because Italy crashed into Africa, and the marble that you have there, the Carrara marble, is just a piece of smear. So it's hard to survive for a fossil that long.
I think about
Shit happens.
the what are they called, like fossil shows. You think about, are there potentially any like transitional fossils that accidentally end up in somebody's private collection and they have no idea what they've got, and
That's the
like
other thing. Think about all these dinosaur species we described from early 20th century bone collections or fossil collections. It's very expensive to make a dig out, get money to go and get yourself a sunburn somewhere in Arizona. Surprisingly expensive, not to mention collections in Mongolia or Africa. So Spinosaurus, that famous thing that people are so excited about, the original type specimen for Spinosaurus was in Berlin. It was discovered in a German colony in Africa. Meanwhile, it was being kicked out of Africa by the British. Then the British got kicked out by the Africans. And Berlin has been bombed in World War II, and that type of specimen of Spinosaurus has been pulverized.
Right.
Meanwhile, we found new ones. It's very expensive to make such a fossil collection. And so we have more findings in all those colonialist drawers were collected earlier. And yeah, it might absolutely be that the key species of, say, transition of flowering plants, or the key species of the transition of birds, or whatever there is, sits somewhere in a drawer catching dust and hasn't been found yet among the rubbles that late Professor, what's his name, collected back there in Cameroon when Cameroon was still a German colony. Things like that. You never know. Namibia, Dutch Südwest, as it was called. So you never know.
Yeah. In Wonderful Life, Gould talks about how Conway Morris, who's one of the paleontologists who studied the Cambrian, he did a lot of his studying of the Cambrian explosion by going through drawers at the Smithsonian. Was just opening them up, going, "Oh, look, there's a new fossil," doing the monograph on that one, and going to the next drawer and just going like that, finding all sorts of new, undescribed species that had been sitting there gathering dust for several decades.
Mm-hmm.
Fun times. But enough of the uniconts. Let's go down the biconta group. Which is the algae and the plants, or algae and plants, because there are lots of, as we say, the red algae, green algae. Then there's brown algae, which isn't actually part of that group; it's part of a different group. There was a recent paper, I think by Andrew Knoll, came out just last year about the evolution of green algae, because that's a very fascinating group. I actually had Matt Heron on, I think it was about a year ago or so, who he studies Volvocene algae, and his research is in looking at the evolution of this group, because they kind of have the whole spectrum of multicellularity in there.
Sometimes independently, yes. The Volvox group the chlorophytes, different ones have different types of multicellularity. And then also you have other algae which make like large cells with many nuclei in it. So like Chlorpa is in that group. Then another one is the Cladophora algae. Nightmare of my students. Sometimes to my poor students, I have to look at Cladophora and they are puzzled what they look. But yeah, there's different types of multicellularity that evolved in the green algae group. And again, when we talk about algae is like Protista. It's more a functional name.
Mm-hmm.
It's these water-dwelling, non-vascular plants. The non-embryophyte, non-vascular plants living in water. Or organisms that do photosynthesis. Can't even call them plants. So yeah, algae is a very broad expression of a group of organisms that have a similar lifestyle, but they're not always necessarily close related. So let's leave the brown algae and the diatoms and dinoflagellates away for a moment.
Mm-hmm.
That's a different group. They live in water, they do photosynthesis, people call them algae, but they have not particularly much to do with green algae or red algae. Green algae and red algae together, they belong in the group called Archaeplastide, archaeplastids in English. Now, arch or archos means senior, if you really translate it. It means also ruler. But it's more like in the sense of ruler because he's the old one, he's the elder one. And in the old times, the older one rules. It's always that gerontocratic system. And so the senior is also a ruler. And so if you have an archbishop, you have the senior bishop, the top bishop. If you have an arch enemy, that's the enemy that has been with you forever and it's your nemesis, it's the one that you hate the most, like Joker to Batman. These are two arch enemies. And they have been enemies for a long time, forever-ish. So again, senior enemy. So the Archaeplastide are the senior plastid carriers. They're called like this because the second endosymbiosis, the invasion of cyanobacterial relatives into eukaryotes, bringing the green photosynthesis into eukaryotes. That happened in that line, and apparently once.
Mm.
So other photosynthetic organisms such as brown algae or diatoms, they pick then this photosynthesis system from a archaeplastid secondhand. Kind of bacterized by snatching the chloroplast. So in that group, that is what the green algae, red algae are. And I would rather focus first on that group. And probably you want to go towards land plants, right? That's where you aim for. Huh?
That's where we want to head.
Good. So that is up on that green algae line now. We go up on the green algae line. So we go up to the base of the Archaeplastida. The chloroplasts have invaded. Now we branch off to the red algae. We leave that to the side. We ignore the red algae. We go to the left. We go to the right. Follow up on the green algae clade now. All right. So we're in the green algae. And there we go. At the base, we have groups. Something that's called, spell that, Chlamydomonas. Chlamydomonas in English, Chlamydomonas. It's little single-celled organism, very simple lifestyle. And that is basically a prototype of all green algae. From that, you go to your volvox that you talk about. Volvox is basically a colony of Chlamydomonas. You can go to ulva, which is the sea lettuce. You can go to Eulotrix, that's the harbor maidenhair algae, that long.
Spirogyra.
Well, that's the other line. You're going after the thing. And then,
that's right.
That line, you go into the so-called streptophyte, which contains Spirogyra, and eventually land plants. Yes.
Yeah, and it seems it's very interesting. Oh well, I do also want to ask you, what do you think about the blue-green, or what are they called? The glaucophytes. Are you familiar with the glaucophytes?
Yeah, it's just a side group of the Archaeplastida. I don't know where they bounce out. I don't exactly know. I dislike the sister to the other Archaeplastida, but I don't deal with them, and I don't have an insight of what role they play for us. So I haven't looked at them pretty carefully. I'm sure about them. But
Yeah, it's kind of interesting. I'm very interested to see where they fall out too, because there's only like a few known, just a handful known species, and because you have the big group of green algae and the big group of red algae, but then just a teeny tiny group of the glaucophytes. Uh, I don't know. It'll be interesting, I guess. But yeah, the the green algae seem almost indecently eager to evolve multicellularity.
Well, as I say, several green algae do that, and there are different ways how you can get multicellularity.
Hmm.
The most common one is going to a filament. And filament is realized also in brown algae and red algae and in even diatoms. It can even be filamentous. And filamentous alignment is simply that when you divide, don't lose contact. Stay connected somehow to your sibling cell. If you always divide in the same direction, you will elongate. And as long as you're filament, you just keep dividing, there's nothing stopping you. If you then divide perpendicular to the previous division sign, you form a side branch. Again, you just continue this. It gets much more complicated if you want to form a two-dimensional or three-dimensional tissue, a so-called parenchyma. Then you get into a conflict. If you divide one cell, not the other one, you get a space issue. Suddenly you have two cells in line with one cell next to it. How to elongate the other cell or divide it too. Then you need to concert what they do. That makes life a bit more complicated. And so it is very interesting to see which of these green algae have developed two or three-dimensional tissues. And it comes surprisingly late and surprisingly rare. So ulva has a double two-dimensional line. So it has two layers of cells that just grow in an infinite long plane. That's coelatus thallus that people can eat. You can eat that. And then it's pretty much it. And the Cladophora and Caulerpa group, they form similar to Acetapellaria, the mermaid's cup, large single cell. Now in Acetapellaria, you stay with one big, huge cell with one nucleus. It's several inches long, forms a little mushroom-like head. It's all one cell, one huge, big cell. And probably gets to the border how large one cell with one nucleus can possibly be before you break down physiologically in your entire DNA management. And so things like Caulerpa or Cladophora, they have large cells that reach almost macroscopic parts, or in Caulerpa, very big actually, become really, that's this aquarium algae that people have in saltwater aquariums. They're pretty large. It's all basically one big cell with many, many, many nuclei in it. It's not really multicellularity in that sense as we understand it, but it is the same effect. You have a large body, and you supply that large body with enough enabling nuclei, except that these nuclei are not separated into single cells, but they swim around in one big cell space. And then when you divide that, subdivide it, then you get the classic multicellularity, which you actually find only in a non-filamentous lineup, in a at least two-dimensional lineup, at the very end of the streptophytes in Coleochaete, which forms a flat, little tiny disc, less than a millimeter large, sitting on other algae or stones, before it goes into a filamentous pattern. It's a little coin-shaped disc that grows like a sucker on surfaces before it then goes into filamentous stage. And that teeny-weeny two-dimensional thing is not really what I would call an impressive multicellularity. There's also not much cell function division, but then when it goes into the filamentous stage, there is cell function division, and some cells will be reproductive and others not.
Mm-hmm.
Not everybody has enough food and can go lucky.
Right.
And then real differentiation happens only in Cara, which is also called the stonewort or moose grass, and some other expressions here, that has a bundle of 6, 7, 8 or 10 cells in a row, they can form little branching nodes or longer bundle sticks. Some of those little branches, they form then reproductive structures like an egg carrier or a sperm carrier, which are actually miniature side branches. They kind of take a different route. Instead of becoming long branches, they stay short and fill the central cell with a lot of food that's in the egg. Not very impressive, but one big thing that changes that in this Colochaeta and Nicara, only the edge cell will divide, and the other cells just mature but will not divide anymore unless they're triggered to divide, unless you tease them. But growth happens only at the end. Pretty much that's it, the multicellularity. So cell division in a tissue is not very impressive on the landworts land plants.
Mm-hmm.
And then even very primitive land plants, very primitive things like Rixia, fluitans, the little liverwort, Rixia liverwort, their body is not particularly impressive. They have multicellular bodies that grow within the apical cell and several layers of cells, but it's not something to know about, a big deal. So yeah, so multicellularity came only later than as the land plants had to deal with a more complex environment. So multicellularity in algae has evolved several times into splendid things like brown algae kelp. But in green algae, it's not so simple and hasn't led to what people would believe as multicellularity as such. Cooperation of cells in a group, yes. Volvox does it already, and some filamentous algae do it, and some have simple thallus, but it is not too much of a complexity as we compare it to land plants, then yes.
Yeah. Speaking of the land plants, which side do you fall on of the bryophyte controversy? Are they paraphyletic or monophyletic, do you think?
So the leafy mosses, so the non-vascular land plants, what people call mosses, falls up in three major branches.
Mm-hmm.
So-called liverworts, the hepatophytes. Then you have the hornworts, which are not so many species and hard to find. And the third one are the leafy mosses, which people know as moss. That grows on trees, they're kind of green pillar. These are leafy mosses. And these leafy mosses themselves, they fall again in three branches. One is the peat mosses. I'll just bring two here. The base of the peat mosses that we know from peat, sphagnum mosses. They've been around for a long time. And they have some really old-fashioned features, like when they come out of the spore, they look like hepatophytes or like algae. They're not very. That it takes them a while to look like a real moss. They also have some old fossils. Then you have on the other side a more sophisticated leafy moss, which are very often called the peristomate mosses. And those peristomate mosses, some of them, they have in their sporophyte, the diploid generation, they both have a haploid generation. That's the actual moss that's here, and the diploid generation, which is just this little stick sticking out, which is the sporophyte. That sporophyte is corresponding to what you see in a land plant. So if you look at an oak, you look at the sporophyte. If you look at a fern plant, you look at the sporophyte. That tiny little stick that lives on a short time in mosses is actually homologous to the big plant of an oak or of a fern. So this sporophyte, where does it come from? You look at the peristomate mosses. Inside of the sporophyte, you have a conductive tissue that allows in peristomate mosses that sporophyte to grow longer and to develop on the top a bit later than in other non-vascular land plants, where that development has to happen much earlier while you're still connected to the base part. Because you can pump food and water off. When you look at the microsection of this, the microstructure, they look in the anatomy strikingly similar to a vascular strand in vascular plants. The cell build is the same. The arrangement of those cells is the same. The only thing that seems to be missing is lignin, that little resin-like wood struck thing that makes the combination of cellulose and lignin is wood. So lignin is missing, and that is what gives stability and rigor to wood. If the chair would just be cellulose, it would be as soft as a piece of cardboard or a shirt. It wouldn't carry you. So theory, the controversy is now this. Is this peristomate sporophyte with this vascular tissue the ancestor of the vascular land plants? Or did that evolve 2 times independently? The catch is this: if it is the ancestor of vascular land plants, then the mosses split up and become paraphyletic.
Mm-hmm.
All mosses that are not peristomate are a gradual stem group, paraphyletic stem group, then something out of these peristomate mosses is the ancestor of all vascular plants, which would mean that we have to split down all leafy mosses. Or if it evolved independently, that the sporophyte developed a conductive tissue, and then on the line to vascular plants, their sporophyte developed a conductive tissue, very similar in convergent evolution, except that in vascular plants it added lignin. Then you are monophyletic with the mosses and monophyletic with the vascular plants. And so that is that debate. Where do I fall to? So first I have to explain what is the debate.
Right.
Second, which side do I fall to?
Right.
In my emotions, I fall with the idea that this sophisticated vascular system, or this conductive tissue in peristomate mosses, is so similar in terms of cell function and self-specifications that I would believe spontaneously, impulsively, that it is homologous to the vascular system in vascular plants. Only lignin had to be added, so just add lignin. Recent publications for mostly Chinese authors, I cannot pronounce at this moment, cannot recall them. Sorry about that.
That's all right.
A lot of that comes from China recently, from Chinese publications, indicate that it is convergent, that the moss conductive tissue is not strongly homologous to the vascular land plant conductive tissue, which gives me a long face. Yeah, exactly.
Sad face. I don't blame you. Yeah, that was.
I always tell my students, I don't teach you my beliefs. I teach you what the evidence says. But put that in your side burner or your back burner. Maybe one day somebody will come, we got to change that story. Then you will know Dr. Buzgo was right.
Well, you have the same theory for the other controversy, what you call the Buzgo heresy.
The Knetalian heresy.
Yeah. I don't know. I like Buzgo heresy more, but yeah, the
no. It's much older than me. It goes way, way back to other people. I'm just a heretic believing the heretic beliefs, an apostate of the monophily of gymnosperms.
Right. The other term for it is the antophyte hypothesis. That's the other common name, yes. Which we did do a video about the evolution of flowers, and we discussed a little bit, and we really just kind of went over it. Just basically, here's one hypothesis, and here's what the consensus is on it currently. But tell us your thoughts about the antophyte, because I know you told me before your line of reasoning as to why you hold the position you do. So if you would like to...
Well, I hold the position of the textbook.
Well, I mean, when you're in the classroom, I'm saying otherwise.
And when I put my black cloak on and go into the occult temple, where we have candles holding upside down and mutter strange wokes that I
all the heretic botanists get together.
Heretic botanists, yeah, the conspiracy of the heretic botanists, yeah.
Like that could be a movie or a TV show.
It could be something, yes. So, the thing is, for people who are not familiar with this, when you look at seed plants, then of course everybody knows flowering plants. And flowering plants, they have the flower as a cone, as a strobilus, as they call, is a cone, a short shoot of leaves packed densely together. These leaves are, of course, the petal, the sepals that you know from roses and flowers, but then also the stamens. And in the middle, the carpels. These are those leaves that enclose the seeds. That's the flower, and it's always this way. You're originally bisexual. Some flowers gone unisexual in evolution, like oak flowers or flowers and hazel flowers, and some pumpkin flowers. They're unisexual in function, but they start as a bisexual cone, and they simply suppress the male or female part, and then are functionally unisexual. Plastic flower is a bisexual structure where you have male and female organs in one close proximity. And also you have your ovules, your seeds, wrapped up inside of an additional cover to protect them from outside hazards such as insects, as the carpel. And inside of this carpel, these ovules, they have again two covers instead of only one, and integument, which is the seed shell. When the seed is ready, that is part of the seed shell. And they have two, an outer integument and an inner integument. So two integuments, a carpel and a bisexual cone. Just look at this in a moment. You have then the other group of seed plants, which includes the conifers, the pine trees, the spruces, but also ginkgos and cycads. The people in America call them false Sago Palms, and nowadays they leave the falls away and they sell them at Lowe's and Walmart as Sago Palms, which is completely wrong because there's actually a true Sago Palm. I think Sago is the real Sago Palm. It's a flower.
Probably have the cycads in our backyard. They call them Sago Palms, though.
Yeah, yes, that is, so these cycads or false Sago Palms, as they call them here in the South, they're gymnosperms. They go back to the Carboniferous time, long time before there were flowering plant fossils around. For a long time, it was believed that first evolved gymnosperms with naked seeds. The seeds are not covered at the time of pollination, like in conifers or in ginkgos. Ginkgos are very obvious. The seeds are basically free dangling when the pollen flies at them. And then some of them wrap them up in additional covers to protect them, and that is where the angiosperms evolved from gymnosperms, the flowering plants. Mm-hmm. When you look now at the angiosperms as different from gymnosperms, it's of course not just the carpel that is different, it is also that gymnosperms normally have only unisexual reproductive cones. They have male cones and female cones, and very often, like the ginkgo and cycads, they have even male and female plants. They're not even the same plant. So they're absolutely not bisexual. They're not bisexual. Then leaves differ. The leaves in gymnosperms develop in a different way than in flowering plants. Classic leaf in a flowering plant is something what you find on a magnolia, for example, or a fig tree. They grow at the edge of the leaf and make this network of veins in it, and if you ever look at a ginkgo leaf, they don't have that, and conifers make just needles. It's not the same thing. There's some other differences. In genes, genetic parts, so the flowering plants, they obviously came from something that looked like a gymnosperm, and people would naturally say it's a gymnosperm. But what happened is now that when people looked at gymnosperms, they found actually one group of gymnosperms that actually have a lot of things in common with flowering plants. They have the same leaves, like flowering plants that look almost like pepper or magnolia leaves. They have bisexual cones, where male and female structures co-occur in the same cone. They don't have a genetic separation of male and female, like other gymnosperms. They have only one gene that starts this cone formation. They don't have one for the female cone and one for the male cone. They have only one. And later have to differentiate male and female functions somehow else. The same thing that happens in flowering plants. Besides this, they also have a double cover over their ovules. Mm-hmm. They don't have only one integument, but they have a pair of leaves that cover the ovule on top of that. We do know that the outer integument of flowering plants used to be leaves. while the inner integument not.
Right.
Distinct origin from the two. Although they look very similar, they have a huge time difference when they evolve. So they have a lot of things in common with flowering plants. Another thing that you find in flowering plants are vessels, highly efficient saline elements, so water pipelines that are continuous and broad. You don't find this in gymnosperms except the Natalians. They have that too. It would be a very, very big coincidence if all of those features, which some of them are not functionally interrelated, happened in the same pattern twice independently.
That would be something.
That would be something. And so I would say it is absolutely possible that Natalians are the ancestors of flowering plants. But then I have DNA against me. The evidence speaks against me. You do a DNA analysis, you see that the flowering plants pattern out separate from the gymnosperms, making them both monophyletic. And then Natalians are sitting in the middle, embedded in the gymnosperms next to the pine family, the Pinaceae.
Right.
Wow, now I have evidence against me. But then there is one thing that makes me go, You look at phylogeny of this Netum group, have an extremely long branch. They kind of literally stick out from all the other gymnosperms. A lot of changes must have happened in those Natalians. They piled up over time, creating these large branches with many changes on it. We do know from fossil records a lot of Natalian relatives died out. They didn't look like Natalians in the first place. Some of them look like cycads, but they're Natalians. So a lot of these things died out. We have a huge loss of species. We do know from phylogeny analysis, undersampling of species causes what's called long branch attraction, that lines of a lot of changes embedded in another group break off and are, by the computer algorithm, planted outside of that large group in which they would have been embedded. You can break down such false long branch attractions, such artifacts, by sampling more species that sit along that branch that help you to embed in the right place. The problem is the Natalians have died out of a large group. There's only 3 of them surviving: Netum, Welwitschia, which is a very strange plant, and Ephedra. So Ephedra and Netum being the 2 key representatives of that line, with Welwitschia being kind of an odd plant morphologically. That is not a good representation of a group that used to be as big as the rest of the conifers, possibly, and very, very, very successful in fossil records just before the flowering plants arrived. That's the other thing. The fossil record shows us a broad diversity of Natalians just before the flowers arrived. And so that long branch indicates to me that no matter how much genetic support we find for a monophyletic group of gymnosperms with the Natalians embedded and the flowering plants separated, as long as we don't get species sampling, we can't be absolutely sure that this long branch is not the beginning of a much longer branch where at the end the flowering plants would stand. And it's simply the algorithm of the phylogeny analysis pops the flowering plants off that system and plants them separately as a monophyletic group. That is more antified heresy. But of course, I'm not the first one to say that. Not one. And that's why I don't call it the Buzgo heresy. I call it the heresy or the antified heresy.
Well, here we will dub it the Buzgo heresy on this channel. So if we had this conversation before I did the flower evolution video, I could have put that in there. And now we transition to the Buzgo heresy.
Well, you would have to. What would be really cool if we could, again, your time machine, and we could go to the Cretaceous time. We will find a lot of plants that are in the Natalian group. We'll be able to collect DNA and then do the DNA analysis. The sad thing is that from petrified fossils, we cannot get DNA. We get DNA from very, very old fossils already. that are not petrified. But we can't get it from completely metamorphized, petrified material.
Yeah.
So if all that we find is a piece of coal between two stone slabs, we can't do the DNA analysis. But I'm not desperate, because we have found very, very fine biomolecules in very old fossils, down to the point of recognizing hormones or pigmentation in dinosaurs.
Yeah, there was oleanine, I believe is the name of the chemical they found in a couple of groups that are probably closely related to the flowering plants. Because I think it is oleanine, if I remember correctly.
Well, as we know with chemical compounds, interestingly, they can occur in a lot of different groups. If you
Sure.
think about salicylic acid or caffeine, it's not just coffee making caffeine.
Mm.
And salicylic acid, also known as aspirin, was described not from willow, salix, but from spirea, which is a not close related member of the flowering plant. So chemicals could be DNA, but we don't know if one day we were able to actually reconstruct DNA from fossils that have been petrified. As I would say this, we have been able to reconstruct the body coloration, the whole body coloration of a Psittacosaurus, those little ceratopsid dinosaurs. One of the questions was whether they were bipedal or walking on four legs, and detecting that they had actually a dark colored chest but a white tummy below, white abdomen, indicated these animals actually indeed walked bipedal very often, because dark chest but white tummy makes only sense when your upper body is higher up for a while and does not play that camouflaging contrast situation. So if you would have a wide chest, that would indicate that you're more walking on four legs and have that part facing downwards. But if it's brown, it indicates it was more lifted up and doesn't make a white flag telling the carnivores where you are.
Yeah.
And so,
There was
so if we are able to do that by now and pin down, for example, coloration of microstructure in feathers in Velociraptor, not Velociraptor, in Microraptor, we know that Microraptor was a black bird-like organism.
I actually met the paleontologist who was on that team, Dr. Julia Clark.
Excellent.
Yeah.
There you go. If you can look at the microstructure of feathers and identify that those feathers were not just feathers, but they were actually black feathers, and you can look at with a blue hinge on it, and you can go to, maybe one day we'll be able to reconstruct enough of the DNA that we have in these plants to be able there to tell us where they actually belong. So maybe one day we'll have some of these Nittian fossils.
Yeah, maybe they're on like an island out in the Pacific somewhere, and we just haven't hit the island yet, or something. You know.
Maybe, or we find a fossil that is not completely petrified, but is simply some smudgy grass or smudgy leaf between wet clay. And I have seen magnolia leaves being analyzed that were exactly there. There was clay stone, and these magnolia leaves were baked into that ancient clay, like in mud. And it was possible to extract DNA from those magnolias. Now, they were not 60 million years old or 100 million years old. They were only about, I think, 20 million years old, but that's already quite good.
Yeah. There was one other thing on the coloration issue. There was a discovery a couple years ago. I don't know if you're familiar with it, but this little theropod dinosaur, which had feathers, also from China, called Sinornithosaurus, had a tail apparently like a raccoon. It alternated between red and white, or sort of like a reddish brown on the tail, red and white. Yeah.
Oh, or like a little panda bear, like that, like the ginger panda bear, the ginger raccoon, the little panda has this red-white ringtail.
Yeah, it was it was kind of neat, I thought. But that was just another point to that example, or another example on that point.
Yeah, and 30 years ago, there was not good enough. We were not at a point to kind of recognize such details. So maybe 30 years from now, we will be able to actually, from some way that I can't imagine at this time, be able to identify at least portions of the DNA of Asian plants, or we find fossils by sheer luck that have not disintegrated to such a point. Kind of like the Denisova, you found the Denisova hominin once. You could use a toe bone to analyze it, but it yielded so much information for us. We don't quite, we can, from the genes, reconstruct how they look like. We don't have enough bones to know how they look like. But
Right.
it yielded so much information to us just from a toe. Maybe we find a toe bone of sort of an old Natalian relative, and then we can break down that long branch. But at that time, I'm just a heretic.
Right.
Schoolroom, I will tell my kids what the catechism is, and then at night I will go into my occult little crypt. Heresy.
Right, and right, continue with the heresy, right?
Continue with heresy. Yes.
Well, I believe we've been doing this actually for about 2 hours now. Is there anything you want to add? I haven't seen any questions from the live chat really. We did have 15 people watching.
15 people are watching. That's incredible.
So are there any concluding points that you want to add?
Well, it's hard now. No, I don't think so, because we have run through the entire, basically, short story about the entire life on Earth from the beginning of with flowering plants. I thought we would split that down to little bits so people can listen to, because I don't know who's listening to 2 hours. So I'm always worried about.
We do have people still listening.
YouTube's are for me more like 3-minute gig. That's what I'm so amazed about. These people, I'm very happy to hear this, but I did not expect that. So in that sense, I think all these sections that we visited, there's a huge story behind this that we are not able to expand in these 5-minute gigs or these short visits we can give each chapter. And one of the parts is that these scientific papers are very hard to pin down. They're hard to access, and most people don't read them.
Oh yeah,
and
absolutely.
I find it amazing how much is discovered, how much we know already, and how hard it is to maintain that knowledge altogether in a world where on one side we have politicians who are anti-science, and on the other side, budget cuts cut into your possible funding, and on the third side, there is a considerable amount of people who struggle with the time to learn to read things like this, because they have to run 2 or 3 jobs at the same time. People who have to run 3 jobs can't follow. They have to run jobs. And I think I see a little bit of a worry about this. And the entire movement of a society against science that we see, I think, is a consequence of that fission between large groups of the population and the scientific community. The scientific community is spearheading, but the staff of the spear has fallen off. You are the spearhead, you're the blade, continuing to fly into the future, but the stuff that supports you and that makes you having the thrust the spear needs to have and to continue is getting thinner and thinner. And I don't know how long this can go on like this. And that is one of the worries I have. And that's not just for evolution. It is also for languages, understanding politics, understanding finance, understanding historic context, where I see the same thing happen, not just in biology. And I'm really worried about this, where we're going to lose a large group of the intellectual capacity of the people, not because they're lazy, but because they can't afford to follow up with what's going on. I don't have an answer to that. I don't have an answer to that.
Yeah, I think you're right. It's kind of scary. But there is hope also. We get people interested in science. Hey, I think this would be a time, I figured you would say, take my class, or something. You'd plug your class. I figured.
Yes, go to universities. I don't know where these 50 people are that are listening to us. They might be in Hawaii, they might be in Alaska. It's I can imagine it's winter. You're sitting in Alaska, nights are long, winters are cold. Videos. The thing is, one of the things the internet is pushing very much about. This to knowledge in the internet is an enormous gift that we can employ today. Did you catch that, or did you freeze off? You read again.
Oh, it's I caught part of that. Oh, it says it's red. It's not changing. Well,
White again here.
it is okay. Well, it says they're connect. Oh, there it goes again. Okay, or it's trying to fix itself. You're right. Yeah, we got to get people educated. We got to get people interested in the subjects. I think a lot of people find approaching science daunting because there's so much jargon and so many different avenues of research, and a lot of it is hard to understand or it's inaccessible because of paywalls and things like that. So.
Yes, and old textbooks. I would like to change the jargon, but then you need to have an agreement among scientists, and that takes an enormous amount of time. And I think that would be something to simplify the jargon in science would be helpful, but then you lose all your tradition background. And that is sometimes necessary, and that is something where scientists should have maybe to, I don't know how they come together because they're, there's a lot of knacking and gibbering on between them instead of finding a conclusive jargon. But yet that would help a lot. But even if you look at other things, as I mentioned, it's not just science, it's also history and politics and finance. Everything has jargon on itself.
Sure, yeah.
And so all I can do is hope that people find the time to educate themselves and not waste time on things, not naming any names here or exciting or entertainment things, but they're really not helping you to get any smarter at all. They are not intellectually. That is something where I think I just can hope that people manage their lifetime good. I tell my students, you're 20, you've already spent a quarter of your average life expectancy. So people who are 20 have already lost a quarter of their lifetime. At my age, I'm already beyond half, way beyond half. So time is ticking off, and our time of life needs to be spent very wisely. What we want to do with our lives. There's things which are more interesting and more fruitful than others. I hope that people put the time together.
Absolutely. Well, I do want to thank you for coming on. Thank you so much for coming and sharing your knowledge.
Thank you for having me and allowing me a space to expand myself here. So, thank you very much.
Hey, I've been excited for this to happen. You know, we got all the questions hammered out and the time and date and everything, and
you will have more sessions like this with other people and other exciting topics, and maybe me one day again. So we will rotate this, and you will have exciting discussions every week, right? Or every week.
It's more like once a month, given school and work. But and plus you got to get and then you got to get your time schedule, and you've got to get their time schedule, and so you got to work all that stuff out. I don't know. Maybe I could have you and Matt Heron on. That'd be neat. You guys could talk about algae and stuff.
Algae, sometime, yes. Algae and
Because he's all into Volvox and stuff like that. So that would be pretty cool.
Yeah, he probably knows much more about than I can. I just know the general part about them. That's going to be very interesting to listen to him. Yes.
So
when you have him on the channel.
Yeah. Yep. I do have his email. So food for thought.
Food for thought.
So all right, everyone. Well, if you could stay there for just one moment after I close out. But
yes.
Thank you everybody else for watching. You have a nice night, and we'll see you next time. Bye, everyone.
Bye-bye, everybody. Happy weekend.